Hello, everyone, and welcome to this audio podcast today. This audio podcast is part of the Cytometry Cyclone. This audio podcast is part of the Rakas and Bombard. This audio podcast as part of the Rakas and Bombard episode, and this audio podcast as part of a Storm Lobostics production. In this audio podcast, and part of some works looking at looking at aspects of immunobiology, and looking at looking at aspects of serology and laboratory medicine, and where as part of the series we'll look to debunk some of the challenges in around some aspects of human transplant immunobiology, sort of more generally. In this audio podcast today, to begin with, looking at the intro and T cell activation, allo recognition, and allo response in human renal transplantation. So initially, it is important to begin with the basis of self for T cells. So as early as 1975, as early as 1975, author and investigator Zinkernagel and Blandon had informed that self consists of peptides only identified within the thymus, and that all other tissues are ignored. Works from investigators Doherty and Zinkernagel in the 1970 and 1975, then also informed that self consists of the major histocompatibility complex, or the MHC, and additional peptide complexes. And so for B cells, author and investigator Cohn sort of earlier sort of defined a self as a cell surface and soluble molecules, postulating that housekeeping antibodies to intracellular components might help clear cellular debris. Ultimately, immunology and context of immunology and the immune system has a basis wherein homeostasis exists through a certain subset, wherein pathophysiology is as self, and a particular fraction of the rest of the immune system as foreign. Specifically, the immune system can be sort of contextualized into four aspects. Firstly, visible self, looking at bodily structures to which the immune system is tolerant. Secondly, sort of the invisible self, bodily structures to which it is not tolerant, but to which it is not normally, but which it does not normally respond. Thirdly, visible non-self structures to which the immune system normally responds. And then fourthly, the invisible non-self, which the invisible non-self and non-immunogenic structures to which it does not, non-immunogenic or immunogenic structures to which it does not respond. So one of the main challenges at the same time of this underpinning of immunobiology is that it is challenging to completely discover the rules by which it has its own sort of own sort of sort of characterizations. So, for example, works by investigators toward the mid 1970s, in 1974, Waldman and Monroe, and specifically or particularly Waldman identified that self T cells consist of common peptide MHC complexes, thus beg the question of what happens when, during a normal life, peptides change. So, as informed by Zinckernagel in 1970 and 1975, placid T cells are often activated by cross-reactive environmental antigens, and autoimmunity then becomes an occurrence when there is cross binding. In self and non-self models, T cells must be able to sort of distinguish the difference between foreign and self antigens. In an immune system poised for danger, T cells need only to identify antigen presenting cells or APCs from everything else. Specifically, if any T cell specific APC antigen presenting cell antigen were omitted, and if APCs were the only cells to activate T cells, the only T cells that are stimulated during an immune response would be those specific for antigen presented on by a antigen for new antigens presented by antigen presenting cells. So the initial recognition of processed antigen by T cells is through a T cell antigen receptor, and accessory molecules interlink APCs and a T cell, thus leading to a stronger cell interaction. So, for example, CD4 binds to the constant region domain of class 2 MHC molecules, which CD8 being CD8 for cytotoxic T cell binds to class 1 MHC molecules, and CD4 positive cells or positive T cells, that is CD4 for CD4 being as being T helper cells, can differentiate down at least two pathways to become inflammatory T cells, i.e., for example, Th1 type cells or Th2 type cells, which assist B cells. Th1 cells are also predominantly involved in mediating inflammatory immune responses, though through activation of macrophages, whilst T helper cells, Th2 cells are primarily involved in the introduction of humoral immunity and through the activation of B cells. Th1 type cells also produce cytokines that primarily act on macrophages. So, for example, interferon gamma and tumor necrosis factor alpha, whilst Th type 2 cells are involved mainly in B cell differentiation and maturation, and through, for example, interleukin 4, interleukin 5, and interleukin 6 of the immune system. That draws this initial part of this audio podcast to a close. This audio podcast is part of the Cytometry Cyclone. This audio podcast is part of as part of a Rakas and Bombard episode, and this audio podcast as part of a Storm Lobostics production.